So this is our last lecture of the semester, and in this lecture we'll be covering Chapter 9, talking about DNA-based techniques. The title slide shows a example of a vector map, and we'll be talking a lot about vectors or plasmids in this lecture. What a vector or a plasmid is a circular piece of DNA. Typically, you put these circular pieces of DNA containing a gene that you're interested in expressing into a particular cell line or bacteria strain. And in this case, this vector is made to be transfected into eukaryotic cell lines. Okay, and we'll talk about the pieces, the arrows in this vector a little bit later in this lecture. Okay. But first, before we do this, I thought it'd be helpful to do some review questions. Okay, so the first review question: what's the difference between DNA and RNA? Okay, I think you might want to pause the lecture and try to go through these and try to answer them. The difference between DNA and RNA, let's look at these individually. Okay, so DNA is missing the oxygen at the 2' carbon. That is true. Okay, that's why it's called deoxyribonucleic acid. Okay. Number 2, RNA forms a stable single-stranded helix. DNA forms a double-stranded helix. That is also true. RNA undergoes rapid base hydrolysis. That's true. RNA is less stable, especially in basic conditions. Okay. RNA can catalyze reactions. That is true. So all of the above 5 would be the correct answer for this question. Okay. What is the name of the following base? Okay, so this is might be a little bit tricky. Okay, and this one's actually 5-methylcytosine. Okay, so the counting starts 1, 2, 3, 4, 5. Okay, and that would be the methyl group there. If that wasn't there, this would be what cytosine looks like, cytosine. Okay. So important on an exam. I mean, I guess I would technically I would expect you to kind of know that because you should know what cytosine looks like, and then you can just add a methyl group to it. But I'd probably stick to the more common bases, at least for the majority of the points on a particular problem about structures. Okay, the DNA oligonucleotide, abbreviated ATCGAC, has 6 phosphodiester bonds. Is that true? Well, let's see. There's a phosphodiester bond between every base, so there'd be 1, 2, 3, 4, 5. So that is not correct. It has a hydroxyl at its 3' end. And if you remember, when you see a sequence like this, it's written in the 5' to 3' direction. Okay, so the 3' would be over here, and yes, in fact, there is a hydroxyl at the 3' end of DNA sequences. So that is correct. Has a phosphate on its 3' end? No, that's not correct. We just mentioned it's a hydroxyl. The phosphate is on the 5' end. Okay, has an A at its 5' end, and the base here at the 5' side of this oligonucleotide is an A, so yes, that is correct. So the 2 correct choices here are B and D. Okay. The phosphodiester bonds that link adjacent nucleotides in DNA always link A with T and G with C. That is incorrect. Okay, the A and T and G and C interact via hydrogen bonding on different strands. Okay, so this is asking about phosphodiester bonds and adjacent nucleotides, and so that has nothing to do with these interactions. Okay, are susceptible to alkaline hydrolysis? That is incorrect. Okay, and that's because in DNA you don't have that 2' OH group, so you don't have the same reactivity or ability to hydrolyze those bonds so easily. Okay. Form between the planar rings of adjacent bases. So that is also incorrect. Phosphodiester bonds do not form between the bases at all. Okay, they form between that phosphate group that's on the 5' position of a ribose sugar and the 3' OH group. Okay. D, phosphodiester bonds join the 3' end of one DNA strand to the 5' end of the complementary strand. That is incorrect. Okay, they do join the 3' end of one base with the 5' end of the other, or I should say nucleotide. 3' end of one nucleotide with the 5' end of an adjacent nucleotide in the same strand, not in a complementary strand. So the correct choice here is none of the above. Okay, which of the following deoxyoligonucleotides will hybridize with a DNA containing the sequence AGA, CTG, GTC? Okay, so this one should be really easy, but don't make the mistake of just looking at the complementary bases in this direction. So you might think, okay, that's T, C, T, G, A, and then you would, look down here and find, this matches. You need to take into account the direction this is going. So this is a 5' to 3'. So the complementary strand is going to be 5' to 3', the opposite direction. Okay, so you're going to need to look, basically read this in reverse and do the complementary bases in reverse. Okay, so the 5' end would be matching up down here. So that would be G, A, C, C, A, G, T, C, T. And if you look at that, that would be choice 2, I believe. Okay. And that, again, that's because this is written in 5' to 3'. So 5' on the complementary strand has to match up with 3' of the original strand that we're looking to match. Okay, on with Chapter 9. Chapter 9 deals with DNA techniques, things like what we refer to as genetic engineering or molecular cloning. You use those techniques to in classical biochemistry to express proteins and purify those proteins after that. We'll also talk about PCR. PCR is a very powerful technique. Okay, and there's a few different types of PCR we'll mention. And then there's other techniques that sort of spend a little bit of time talking about, and those are listed down here. Okay. So DNA cloning, also referred to as genetic engineering. What is this? It's the creation of identical copies of a piece of DNA, right? Usually containing some sort of a gene that you're interested in. Okay. You can isolate a specific gene from a source organism and amplify it in a different organism, a target organism. So, for instance, you could pick a gene from a human gene, and then put it into a bacteria cells, and then really amplify that gene product, which is a protein usually, in those bacteria cells. Okay, so the basic steps are listed here. We'll go through this, but really you need to come up with a source of DNA, okay, and that can either be cutting the DNA out of from the host organism's DNA, so either amplifying it via PCR or straight cutting it out. Typically you would amplify it via PCR first and have some sort of restriction site on your primers that you're using that adds specific sites into that. We'll talk about more about this when in our class activity, but it gives you a way to put that piece of DNA into a vector or plasmid right where you want it. Okay, and so that's the next step is selecting a suitable carrier DNA, and those are what we refer to as vectors. Okay, when they have the DNA of interest in them. Okay, then you insert that gene into the vector. You insert the vector into a host cell, and then you let the host cell produce multiple copies of that DNA, or if you're looking to express that gene, multiple copies, a whole lot of copies of that gene product, the protein. So what does recombinant DNA mean? Well, recombinant means it's artificially created and it's combining sequences that don't occur together in nature. And really, anytime let's say you get this is how it usually goes. You get a human gene and you put that into your vector, and then you put that into so right then the human gene in that plasmid, that artificial plasmid, is recombinant DNA, because you're combining DNA that doesn't normally go together. Okay, and what is this done for? Well, cloning of genes, okay, overexpression of proteins. You can also, in industry, you make food that has different genes in it. The only example I can think of off the top of my head is, tomatoes that have certain salmon genes in them to make them grow better in colder temperatures, that sort of thing. And you can even do that sort of thing with animals. Animals that you study in lab are transgenic, and that's where you have you alter a gene of interest in that animal to do some sort of a study. So why do biochemists clone? This is a biochemistry class. Why do they clone? Typically biochemists clone because they want to purify a functional protein to study in a system that makes a lot of a protein. So it's much easier, if you want to study, let's say, hemoglobin, it's a lot easier to make artificial, I shouldn't say artificial, but it's much easier to get enough of your protein to study if you put that gene for hemoglobin, let's say, into a vector, into a bacteria cell, and then grow up cultures of those cells and have them make lots and lots of protein. Lot easier to be able to study it when you do that than if you're trying to extract human hemoglobin from, let's say, like blood or something. You get much more of it when you overexpress it in bacteria cells. Okay, so let's go through the steps of this. Okay, so first, okay, you need to have some sort of a vector that you're interested in, and it's a circular piece of DNA. You have to cut that with what's known as a restriction endonuclease. Okay, or also known as restriction enzyme. Okay. The endonuclease part means that it can cut within a sequence of DNA. You also have exonucleases that cut only from the ends of DNA. But when we talk about restriction enzymes, I think it's usually implied that it's an endonuclease, which you would need it for a circular piece of DNA. Okay, so you cut that plasmid. And this blue piece is, what's taken out. Okay. Second part, you need some sort of a source DNA, and typically it's going to be a eukaryotic chromosome. Okay. And you're interested in that eukaryotic organism's gene here that's highlighted in red. Okay. You obtain that fragment by cleaving the chromosome with a restriction endonuclease, or I think more commonly you would just PCR that out, which means we'll talk about PCR later in the lecture, but it's a way to amplify a certain sequence of DNA, and you can amplify it in a large degree. Okay, so you wouldn't need very much of your source DNA if you use PCR to amplify that product. Okay. And the good thing about PCR is when you're amplifying it, you can put any restriction site you want on the ends of it, so it could fit in exactly where you want it to in your specific cloning vector. Okay, the third part of this, your fragments are ligated together. Okay, so you have to, in this case, this red part is what we're interested in, and the yellow is our open vector. You would put them together with what's known as a ligase, an enzyme that sticks these two pieces together, right? And you would have your recombinant vector. That's your plasmid with that gene sequence inserted into it. Okay. The next step on this, once you have that recombinant vector, is to put it into a host cell. Okay. So you would put it into the host cell. The host cell has its DNA in it as well, and then it will have your plasmid that you put into it. Usually you use bacteria. Bacteria is great because it's a very simple organism. You can grow lots of it. The conditions are very, very simple to grow. It produces lots of protein. But there are other options. Yeast cells, for one. That was probably the first. Well, it was the first eukaryotic system to do these recombinant proteins or molecular cloning, and since then there's lots of other eukaryotic cells that have been cultured. One of them is CHO, which stands for Chinese hamster ovary cells, but there are many of them that you can use that are actually eukaryotic cell lines. Eukaryotic cells are a little bit more difficult to grow, but sometimes what the protein, the gene that you're looking at, or protein that you're looking at, requires a eukaryotic system for things like, I don't know, post-translational modifications that aren't done in bacteria cells. Okay, so after you put that in to the host cell, the next is the propagation, and that's where you just let those cells grow. Okay, and when the cells grow, what they're going to do is they're going to make more copies of that plasmid as well. Okay. So let's look at these steps in a little bit closer detail. So the first steps, the restriction endonucleases, these cleave DNA at very specific locations. They're common in bacteria. That's where they were first kind of isolated from. And what they do in bacteria cells naturally, these eliminate things like foreign DNA. So like infectious viral DNA are eliminated there. Bacteria tend to methylate their DNA, and that kind of signals that this DNA is supposed to be here. And so they have endonucleases that cut DNA that's not methylated. Okay. The picture here depicts some cleavage sites. Okay, so endonucleases, or restriction enzymes, they cut at usually very, very specific sites. And so one might cut at this sequence, shown here, highlighted in this orange and red. Okay, and it doesn't just cut, they don't always just cut, in a straight line. Sometimes they'll cut in a staggered pattern like this. And when that happens, what's left is, after the cut, you have the strands, the ends look like this. They have sort of these overlapping sequences, and what that's referred to as sticky ends. And it's referred to as sticky because once this end sees that end, it's just gonna stick to it, I guess you could say. The other option is at this sequence, so the purpley sequence here is another sequence that a particular restriction enzyme recognizes, and it will cut just straight through in a straight line. So the ends that you'll end up are like this, right? There's no overhangs. So this would be referred to as a blunt end. And if you're interested, the particular restriction enzymes that cut that these sequences are listed here. There are many of these. So you can, if you have to do this, and we'll do this in the class activity, you'll actually look these up and try to find some specific restriction enzymes. Right. The restriction enzymes work by cleaving the phosphodiester bonds. And again, it's at a very specific sequences. Usually they're a palindrome sequence. And if you see here, the GAATTC is a palindrome because its complementary strand is the same, GAATTC. Okay. As I mentioned, some make sticky ends, some make blunt ends. If you're interested, you can look at this website, the New England Biolabs website. They're a big company. They make a lot of these restriction enzymes and sell a lot of them. So that's a good place to look if you ever need to find a restriction enzyme. Okay. Just an example of some sticky ends versus blunt ends, right? So when you look at a restriction enzyme, typically you know where they cut because it'll show you the sequence they recognize and the arrows are where they're cutting. So this would be, sticky end, sticky end, sticky end. This one, however, is a blunt end, right? That's cutting the same position on both strands. And again, here we have another one that would produce a blunt end. Okay, you don't have to know these. I wouldn't expect you to memorize these. These are things that you look up. Just example, one's sticky end and one's blunt end. Here's a crystal structure of the EcoR1 restriction enzyme. It's actually a dimer, and that's shown here in the sort of two different shades of green that are used. What you'll notice is that there's magnesium ions in the active site, and then the DNA backbone shown here in orange. And so the cuts are happening right here, right by the magnesium ions. So these are using magnesium ions to do that hydrolysis. And then you'll also notice one of the reasons why these enzymes typically recognize palindromes, because one of these of the monomers of EcoR1 is recognizing a certain sequence of DNA, and then the other one is flipped kind of 180 degrees compared to this one, and it's going to recognize that same sequence but just in reverse. Okay, so that's why they typically act on palindromes. You can also use synthetic DNA in these vectors, and in a really good use of this synthetic DNA, or I should say, a part of these plasmids that's made from synthetic DNA typically are the multiple cloning sites. And by just synthesizing this piece of DNA, you can add in lots and lots of restriction sites to that. Okay, and then you can put that in your plasmid, and you'll have multiple restriction sites in this very short sequence of DNA. And that's very useful because then there's lots of different options to use, and you can clone in different genes very easily into that multiple cloning site, because there are so many options for restriction enzymes to use. Okay, looking back, we've talked about the first couple steps using restriction enzymes, looking at the third step, the DNA ligase. Oh. So before we talk about that, we'll talk about the vectors themselves. Okay, the term vector or plasmid is used sort of interchangeably. What a plasmid or a vector is circular DNA that are separate from the bacterial or host cell genomic DNA. They can replicate, and they do that because they have what's known as origins of replication in them that the host cell can use to replicate them. Okay. They also typically carry, if you're talking about a bacteria plasmid, they carry antibiotic resistant genes. And eukaryotic plasmids also carry these, what's known as a selection marker. So if you grow cells in the presence of this antibiotic, only the cells that have the plasmid with the antibiotic resistant plasmid in them will survive. And you can clone DNA up to about 15,000 base pairs into a cloning vector. You can clone whole chromosomes as well. And you do that by either BACs or YACs, bacterial artificial chromosome or yeast artificial chromosomes. We won't really talk about those at all in this class other than just mentioning them. Okay, so there are a couple different types of vectors. One is a cloning vector. Those are the simplest. And then another type is an expression vector. Okay. Cloning vectors are used really just to transfer genes, amplify genes. Expression vectors can do that, but they also are good for expressing the gene, so encoding for RNA and protein from those genes. Okay. And the generic vector or plasmid is shown here with some parts to it. The parts that are circled in red are ones that are in both expression vectors and cloning vectors. So a polylinker or multiple cloning site, they typically both have that, and that's where you would insert your gene of interest. Okay, a selectable marker. So if we're talking about bacteria cells, an antibiotic marker, this would be a gene that encodes for a protein that gives this bacteria antibiotic resistance to a certain antibiotic. Okay, and an origin of replication, and that's critical for this to be the plasmid to be able to be duplicated when the cells divide. Okay, so those are in all vectors. Expression vectors also have some other pieces to them. One is a ribosome binding site right before the multiple cloning site, and another is a transcription termination sequence. So why are they there? Well, if you put your gene in there, you want your gene to be able to be expressed into a protein, so you need a spot for the ribosome to bind, and then finally transcription to be terminated. Okay. Expression vectors also include a gene that encodes for a repressor that binds to an operon O and regulates P, which stands for promoter. So O stands for operon, P stands for promoter. And those sequences are in the DNA as well. Okay, we'll talk about that in a little bit more detail. Okay. Here is a plasmid, PET47B. This is one that I've used in the lab, and this is just, if you look, one of these up, what you would see online, a vector map would look something like this, and they have restriction sites listed throughout it. What you'll notice is, there are some restriction sites all over. Okay, but they're really concentrated in this region. So this is the multiple cloning site, and you'll see all the restriction sites that are listed here. Gives you lots of options on how you want to insert your gene there. Okay. Okay. You also have a kanamycin resistance gene that gives it resistance to kanamycin. There's your origin of replication, and finally the LAC-I gene, which is an inhibitor that binds to the LAC operator or operon. An example of another vector, one that I've used in lab, and this is the PCMV6 entry vector. This is a vector for eukaryotic cells, mammalian cells, and it has a lot of sort of the same things. Here's the multiple cloning site. It has a ribosome binding site and some tags on it. In this case, this is a flag tag, or also known as DDK. This is just a tag that binds to very strongly to a specific antibody. So if you want to visualize where your gene that you put in is being expressed in a cell, how much of it, or the location in the cell, that's kind of why that is there. Right? The other part I'll mention here is this neomycin kanamycin resistance gene. Okay, and that's also used to select cells that have the plasmid in them. Okay. The antibiotic selection, things like penicillin, ampicillin will kill bacteria. So if you have a plasmid that carries a gene that gives that host bacteria resistance to the antibiotics, that will give you a way to selectively grow only the cells that have that plasmid in them. Okay. And so the antibiotic resistance gene makes some sort of a protein that either breaks down the antibiotic, or it pumps the antibiotic out of the cell. So in the case of ampicillin, that ampicillin resistance gene makes a protein that breaks down ampicillin. All right, let's look now at a little bit closer at step 3, the DNA ligase. Okay. A ligase is an enzyme that covalently joins two DNA fragments. This is normally functional in DNA repair inside a cell. Human ligase uses ATP. Bacterial ligases use NAD. Some only ligate sticky ends. T7 ligase is an example of one that only ligates sticky ends. Some ligate blunt ends or sticky ends, and T4 ligase is a enzyme that does both. All ligases only work with phosphorylated 5' ends. Okay, so that's important if you're only using one restriction site. And why is that important? Well, if you're only using one restriction site, you have to cut and then dephosphorylate, so you're not just going to stick the same DNA back together again. Okay. So here's a cloning vector, and this is one of the first cloning vectors that was produced. The P stands for plasmid, the lowercase p, BR322. And it's, fairly simple. You have ampicillin resistance gene. You have a tetracycline resistance gene. So that's another antibiotic selection gene. And then you have an origin of replication. And then there are only, 5 restriction sites that are listed here. Okay, so this is what's known as a low copy plasmid, because there's only about 10 to 20 copies per cell, and that's, typically you're going to see plasmids that have a higher copy number, that have more copies per cell than that. Okay. If we look at these, okay, and we just make this a cartoon, the orange is for ampicillin, the blue is for tetracycline. Okay, we have all these plasmids. We can do a certain restriction enzyme, PST1 here, and that will cut in the ampicillin region, the ampicillin resistance region. Okay, then we can add our foreign DNA that has the same ends as that PST1 restriction enzyme. Okay, so why would we do that? Well, if we have a plasmid with two antibiotic resistance genes, what we could do is we could, insert a gene into one of them, and then we can grow these cells in the presence of both antibiotics, and the cells that grew in tetracycline but didn't grow in ampicillin, that's a clue that we have inserted a gene in that ampicillin resistant site. Okay, so when we do that and add that DNA in there, now we can, when you do this, like sometimes what you're going to have, you're going to stick those right back together. Okay. Other times you're going to successfully put in your foreign DNA. Okay. So that, and when you do that, that's going to disrupt that resistance marker, and then those cells then won't grow in ampicillin. Okay. But the tetracycline resistance doesn't change, so all of these are going to be able to grow in tetracycline. Okay. We would then put them in a cell. Okay, those cells now, some of them are going to be able to grow in both antibiotics, some will only be able to grow in tetracycline. Right, so we do that. We grow first in agar-containing tetracycline, right? They're all going to grow. Then we can transfer single colonies from this onto agar-containing ampicillin and tetracycline, or agar containing just tetracycline, right? So we'll take one colony from here, we would put it at the same spot on both of these plates, and do that with many different colonies, and then the colonies that grow in tetracycline but do not grow in ampicillin plus tetracycline, those are the ones that contain that plasmid. And those would be the ones that we would use in further studies. Okay. Transferring the plasmid with your clone or your DNA that you're interested in, that gene. Okay, what's this? There's a couple different names we refer to this as. Okay, one is transformation. When you're putting a plasmid into the bacteria, that's what was referred to as transformation. And you either do this by heat shock or electroporation. Probably more commonly to use heat shock, right? You just put your cells and your plasmid together. You put it in 40 degrees Celsius for like 30 seconds and then take it out. And that's enough of a shock to get some of that DNA into your cells. And those kind of cells, bacteria cells, are referred to as competent cells. That means they can take up plasmid DNA. Okay. If you're putting these plasmids into bacteria via a viral method, sometimes viral methods are used, that's called transduction. Okay, that's probably less commonly used. And then finally, if you're putting a plasmid into a eukaryotic cell, like an animal cell line, this is referred to as transfection. Okay, so transformation, transduction, and transfection really are all the same thing. You're putting your plasmid into some sort of a cell, but the exact cell types are going to be and methods differ in these three techniques. If you want to express those clone genes, and that's the case of biochemists, those biochemists typically want to look at the protein product of a gene. You need to use expression vectors, and those contain sequences that allow the transcription of that inserted gene. And, and again, expression vectors differ from cloning vectors because they have promoter sequences, operator or operon sequences. They code for ribosome binding sites, and they have transcription termination sequences. Okay, and this is, again, we've seen this picture. Okay. Okay, the gene's normal promoter and regulation is not used if this is being expressed in a different organism, something like a bacteria. And that's why you have these built-in promoters and operators to give you the ability to make this in a bacteria cell, where this gene you put in there might be a human gene. Now you can make that human gene in a bacteria cell, and you can have a way of controlling it. And so one way we can control this is by using the lac operon. And you put this expression vector containing your gene into a bacteria cell. You want to be able for that cell to make the protein, but you don't want it to always be making the protein. You want to be able to grow the cells to a certain density. You want lots of cells to be there before you tell them to say, hey, start making this protein. And the reason why is if they're constantly making this protein, they'd be using a lot of energy. They wouldn't be growing very fast, and so you wouldn't get a lot of cell growth, and ultimately you wouldn't get a lot of protein produced. So you want the cells to first focus on growing and dividing, making lots of cells. Then you want to, when they've sort of reached that exponential growth phase, then you want to tell them, okay, start making the protein. Right. And the most commonly used system for doing that is the lac operon, and that's shown here in sort of a cartoony picture of DNA. Okay, what's involved? Well, you have a promoter, an operator, so a promoter sequence, an operator sequence, and then you have 3 genes. LAC-Z, LAC-Y, and LAC-A genes, and a terminator sequence. Okay, what's happening? So basically, what's going on is you're creating a repressor protein with your genes. Okay, that repressor protein binds to the operator sequence, and when that repressor protein is bound to the operator, the RNA polymerase can't get to the promoter, can't bind it, and it can't start expressing these genes. Okay, so the LAC-Z, LAC-Y, and LAC-A gene can't be expressed. Okay. But when you have lactose there, and I should mention here, I didn't put this gene in, this LAC-I gene, that encodes for this repressor. Okay. So this is, always being produced, the repressor that binds to the operator, and LAC-Z, LAC-Y, and LAC-A are not always produced. Okay. So this is the normal, without lactose, this is the operator, the operon is turned off. Okay. If lactose is around, okay, then this repressor gene gets lifted off of the operator, RNA polymerase can bind, and then it can make these LAC-Z, LAC-Y, LAC-A genes, which then go on to they can digest lactose. So this is the normal way this gene would function in a bacteria cell. Okay. What we do in engineered plasmids is we use the LAC-I gene, and we use the promoter and operon sequences. Okay, and then you can sort of envision, instead of these LAC-Z, LAC-Y, LAC-A genes, if you put the gene of interest that you're trying to express in there, now you have a way to control the timing of that expression, right? You can just add lactose to the culture, that will remove the repressor, and then you can start producing your gene. You can start expressing your gene. Okay. Instead of lactose, we usually induce expression from this using a compound known as IPTG. And IPTG is a lactose analog, so it looks very similar to lactose. The difference is it's not going to be digested in the cell, so it just stays around longer. Okay, so we add, we have without IPTG, you have that LAC-I binding the operon, blocking the promoter, and so this yellow part would be our gene of interest. Or no, sorry, it's not the gene, it's the T7 promoter, excuse me. So you would not have access to that when this is bound. Okay, your repressor's bound. When you add IPTG, that removes the repressor, and now that T7 protein can bind there and initiate the expression of your gene product that was inserted after this. Okay. Is protein expressed? How can you tell? Well, in this picture, the first lane is this is an SDS page gel. This would be, the first lane would be before you add IPTG, and the second lane is after. And so in this case, their protein that they're expressing is, something less than 29 kilodaltons, maybe 28, 27 kilodaltons, something in there, 25, something like that. Right, so in an SDS page, as we've sort of talked about before, what you do is you denature the proteins, you mask the charge with detergent SDS, you run it through an acrylamide gel, and then these bands are based on size of the protein. Okay. And I mentioned lane 1 is pre-induction, lane 2 would be post-induction with IPTG. The purification of recombinant proteins, purification of natural proteins is pretty difficult. Recombinant proteins, a good thing about recombinant proteins is they can be tagged for purification. So the tag that's attached to your protein can bind to a specific resin. And most commonly you use his tags. So those are histidines, something like 6 histidines in a row, either at the front of your protein or the back. So the N-terminal or the C-terminal part of your protein, you can add 6 histidine residues, and those bind very well to the nickel 2+ cation. So you have your purification column containing nickel metal ions that are immobilized to your column structure, and the histidine, the 6 his tag, will bind to that and stick to your column. And some other ones are also shown here, but definitely nickel columns are the most widely used because the tag, the 6 histidines, is very simple, and also the nickel column is also very simple. So here's just a depiction of how this can work. You can have your target gene here in red. You can link that to another gene, in this case for GST. And what we call this now is a fusion protein, because you have one protein fused to another via their DNA sequences. Okay, so when these are expressed, you produce your normal red gene that you're interested in, and another protein, in this case GST, here in purple. And those those two proteins now are connected. Okay? And you then kind of go through your this plasmid would be in a cell, and you tell this cell, okay, make a lot of this protein. It does. Then you extract that protein, you break open the cells, and you would pump that mixture of what's inside the cells over your column, and that GST protein can then bind to what's known as the glutathione, which what stands for GST, that's anchored to your column bead. Okay, so that's immobilized in your column. That GST protein will bind to the glutathione, and then you can wash out all the other stuff from inside that cell. Okay, then you would pump in a solution of free glutathione that then binds to that GST binding protein to remove your protein of interest off the column. Okay, and then the other step is usually if you want to look at that red protein, you would have to cut that purple part off. And when you make fusion proteins like this, usually there's a linker between them that has some sort of a cleavable site that you can cut off your tag, the GST binding protein tag. Okay. And how do you assess how pure your protein is? Again, you can use something like SDS page. In this example, what we're seeing is steps along the purification process. This might be your cells after expressing that protein, and then maybe this is the soluble fraction. Then maybe this is your first step. That's maybe second step, and so forth. But then when we get out here, this is probably coming off of your column where you have different fractions, and this would be the protein of interest. And you can see how pure it is because you have one band at a particular molecular weight, and you don't have any other bands at any other molecular weights. Another thing that's often done is when you're studying proteins, you want to not just look at the native wild type protein, you want to add mutations into that protein for various reasons. You want to study biophysically what certain amino acids in a protein sequence, what they're important for. One way to study that is by mutating them. Another reason you might want to add mutations in is for binding particular labeling molecules. Sometimes things will only bind to certain amino acids, so you want to put a specific amino acid in a certain site in a protein that you can then bind a labeling molecule to. Okay. And another reason is also to study mutations that occur in disease states. So if a particular amino acid, if it gets mutated, that might cause a disease in humans, and so you might want to study how is that protein different biochemically than the wild type. Okay. To mutate amino acid, what you need to do is you need to change the nucleotides in the DNA sequence. Okay. Site-directed mutagenesis relies on chemically synthesized mutated primers that are incorporated into a newly synthesized strand of DNA. So you create primers that have this mutated sequence in it, and then you use a technique called PCR to amplify a mutated sequence. Mutated plasmids are always sequenced to confirm that desired mutation is present, and only the desired mutation. By doing this, sometimes you'll actually get more than more than the desired mutation of interest. So you need to sequence the DNA after you do this. Okay, so how does it work? So you have your recombinant vector. You can cleave with a restriction enzyme and put in a mutated vector. This only works if you have a restriction site that's within your gene. Okay. More often than not, okay, that would be sort of an older technique to do this. The newer way of doing that is to have primers that match up with your site, except for these X's kind of depict where a mutation is. So all the other sequence in that primer matches up except that spot at that X. Okay, and so the example here shown is that, so this, you could think of this maybe as your primer. The wild type has these 3 A's, nucleotides in the middle here, and the mutant primer will have GCG, right? So those aren't matching up, right? So you use these, and then you use a DNA polymerase. Those primers will match up. Your DNA polymerase will then copy those strands, okay, and it makes a copy, and it keeps that mutation there when it makes that copy. Okay. Then you would digest the non-mutated parental DNA in blue, okay, and then what's left is your plasmids, but only with the mutations in them. Okay. How do you digest the non-mutated DNA? Well, most bacterial DNA is methylated, so you use restriction enzymes that cut methylated DNA. Okay, and if you do these steps multiple times, cycle these steps, you can make a lot of the mutated DNA. And that's the basis for PCR. Okay. So PCR stands for polymerase chain reaction, and this is a very widely used technique in all sorts of biochemistry, molecular biology, really all of the biosciences use this technique. Okay. And what it is it's a technique that amplifies DNA. You can amplify regions of interest, parts of genes or whole genes within a sequence of DNA. And it can also amplify complete plasmids, so complete circular DNA you can amplify. How you do this is you mix together, your mixture is going to contain your target DNA, primers that are complementary to your target. And so primers are, shorter sequences, something like, 15 to 30 nucleotides long. Okay. You also put in the deoxynucleotides, the triphosphate versions of them. So deoxy ATP, CTP, GTP, and TTP, right? Those are going to be your building blocks to make new copies of your target DNA. And then you also put in an enzyme that lengthens the chain, that creates this copy of your target DNA, and that's the DNA polymerase. That's an enzyme that polymerizes these DNA nucleotides based on a template, which is your target DNA. Okay, so this mixture goes into what's known as a thermocycler, and that's something that controls the temperature very easily, and it cycles the temperature. And the steps are as follows. First, you melt the DNA. That separates double-stranded DNA into single-stranded. Then you cool the DNA, and that allows your primers to anneal to the target. And then you use polymerase that will extend the primers in the 5' to 3' direction. And so that would be, you would raise the temperature back up to do that to the temperature that your polymerase is most active at. Okay, and then you would just cycle the temperature now between these three temperatures. And to do that, every time you cycle the temperature, you're getting another copy of your target made. So a good video to watch is here. There's probably lots of other ones on YouTube that might make it easier to visualize. We're going to go through this just using the figure from the book. Okay, so here is our target shown in blue. Our primers match up with the ends of that target. Okay, one on both directions. Okay, you would so here the first step, you heat to separate the strands. Then you would lower the temperature to anneal your primers to the target sequences. Then you would raise the temperature slightly to whatever the temperature is that the enzyme, the polymerase works at, is most active. That replicates your target, and then you cycle through those steps again and again. Okay, and that's where the PCR comes in. And at the end, what you have is many copies of your gene of interest. Okay, and what's also shown here hanging off of your primers is a specific sequence that recognizes a certain that contains a certain restriction site. Okay, and in this case, it's the EcoR1 endonuclease restriction enzyme that cuts that. Now it has very specific ends, and you can put those into your primer at a very specific location. Okay. Some adaptations to PCR that are not in the 5th edition of the book, but that are in the 6th edition. You can have what's known as reverse transcriptase PCR or RT-PCR, and this is used to amplify RNA sequences. Okay. Why do you want to look at RNA? Well, it shows only the genes that are expressed. So every cell in your body has the same DNA in it. So if you're trying to look and sequence DNA from different cells, you're going to see the same thing in every cell. But the genes that are being expressed differ very drastically from cell type to cell type, and even depending on looking in the same cell type or same tissue, depending on the age of a person, the genes that are being expressed at a given time are going to vary. So to look at that, you really want to look at the RNA. That tells you what genes are being expressed. So RT-PCR, the first step uses reverse transcriptase that converts RNA to DNA, and then the rest of it is like a normal PCR where you amplify that DNA that's converted from RNA. Okay. Another adaptation to PCR is known as quantitative PCR or qPCR, and this is used to show quantitative differences in a certain DNA or certain gene levels. Quantitative PCR is also referred to as real-time PCR because you're getting a real-time measurement of the amount of DNA that's there every cycle. You can combine both of these together to give what's known as qRT-PCR, and that tells you how much of a specific RNA transcript is present in a sample. Okay, very, very powerful techniques that if you go on in biochemistry, you're going to run into, or biology really for that matter, you're going to see these techniques being used. Okay. qPCR, what it's using is fluorescence, and you have a particular probe that matches up with a DNA sequence of a target that you're interested in. And so when the probe is by itself without that target, the fluorophore in that probe is being quenched by a quenching molecule. Okay, when the target's present, your probe is complementary to it, so they can bind together. That moves the quenching molecule further away from the fluorophore, so now you get fluorescence from that fluorophore, so that's where your signal grows in. Okay. And what your signal looks like in qPCR is shown here, right? So you typically on the x-axis is your PCR cycle number. Okay, you do a number of cycles, and your signal, your fluorescent signal, will slowly grow in as the amount of target is amplified. Okay, and it follows this sort of sigmoidal curve. And what you do is then you set a threshold that says, okay, at this point, once the signal crosses this, I'm going to get with a count number, and that's just the cycle at which that signal crossed that threshold. And so for this case, we look at sample 1, sample 2, sample 3. Sample 1 crossed the threshold at a smaller cycle number. That means there's more of that target DNA there because it amplified quicker. Sample 3, it took a lot longer to get to that threshold, so it has the least amount of that target sequence. The last technique that's talked about in the first section of Chapter 9 is this technique that's used to study forensic DNA, and that's the short tandem repeats, STRs. Okay, so these vary between individuals, and they're somewhere along the lines of 4 base pairs that are repeated either between 5 and 50 times. And if you measure, in this case, at least 13 different regions of these are measured for forensic purposes. And you have a dye-labeled PCR primer, okay, that can amplify these certain regions. And these would be your STR regions in orange. Okay. What you'll get out is after the PCR amplification, you get these amplified products that contain your short tandem repeats, and you get lots of them because you're looking at 13 different regions, at least 13, and then you feed them through a capillary gel instrument. And so they go through this very narrow tube that's filled with gel, and so that separates them out based on size. And then when they come out, they go into a detector that reads the fluorescence of them. And what you'll get out are these sort of peaks at certain times. Okay, and in this case, 16 different STR locations are tested. Possible match between 2 people, it's estimated at 1 times 10 to the 18th. Okay, so if you have, if you're looking at these for forensic and forensic data, the likelihood that 2 people would have the same pattern of these is 1 times 10 to the 18th, right? So, 1 in 10 to the 18th. So that's, a huge number. So the likelihood of that is very, very small. And to give you a scale on that, current world population is 7 times 10 to the 9th, roughly. Okay, so you're looking at a number there that's, if you're looking at the entire world population, right, this number is that number squared almost. Okay. This can be done because we know the genome. We know where these STR locations are. Okay. Okay. The last section we'll cover in this class is 9.2, and this discusses genome to proteome. The proteome is really what biochemists care about. What we need, why do we care about this? Well, we're using genes to understand proteins. We want to understand the localization of the proteins. We want to understand the interactions the proteins have. Okay, so protein function can be studied in three levels: phenotypic function, cellular function, and molecular function, or those would be considered like activity assays when you have your protein isolated. Okay. So some protein functions that we can do utilizing nucleic acid methodologies. You can get the sequence and structural comparisons with genes and proteins of known function. So you can look up, you can have a particular gene that you're interested in and compare it to other genes of known function. So say your gene encodes a protein that you're not sure what it does. If you just compare that sequence with other known proteins, you might get an example of what its function would be. You can determine when, where, how much of a gene is expressed, and that technique, qRT-PCR, would be very well used in that. You can also use immunofluorescence and things like green fluorescent protein tags, which is an example of a fusion protein, to visualize where in a cell or really how much of a particular protein inside a cell is being expressed. You can investigate the interactions of proteins with other proteins, and the techniques we'll talk about: co-immunoprecipitation or the yeast to hybrid system. And then you can also do phenotypic function. If you knock out or knock down a gene and look overall, how does that make that cell grow? Okay, so a knockout is completely removing a gene. A knockdown is just making it express less. Okay. So this idea of that we showed earlier in the class, where you have a cloning vector and you can just take a piece of DNA from a eukaryotic plasmid and stick it in there, is a little simplified. Eukaryotic genes typically have both exons, which are coding regions, and introns, which are non-coding regions within one gene. Okay, bacteria can't splice these introns out. In a eukaryotic cell, introns are spliced out. In a bacteria cell, that's not going to happen. So what is the solution? Using the messenger RNA, because that's intron-free. Okay, so messenger RNA is only the coding regions, only the exons. Okay. The first step of doing this is you have to construct your what's known as cDNA, complementary DNA. And it's complementary to the messenger RNA. So if you have a specific messenger RNA for your gene, what you do is you use a primer that sticks to the end of your messenger RNA, and all messenger RNAs have a poly A tail, which is that base A just repeated over and over again at the end of that gene. So your primer can contain T's. So you have a DNA primer that matches up with your mRNA. Okay. Then you would use a reverse transcriptase enzyme to construct your DNA from your RNA sequence. Okay. And so this is also the first step in RT-PCR. Okay, you're creating a DNA sequence based off of a messenger RNA for the gene that you're interested in. Okay, then your mRNA is degraded, and remember, mRNA is or RNA in general is susceptible to basic conditions. So you add a base that removes your RNA. What you're left with is DNA. Then you can amplify that DNA via PCR if you choose appropriate primers. Okay. So that would be how you would get your gene of interest, a eukaryotic gene of interest. Okay, not straight from the chromosome, but using messenger RNA. Okay. Determining protein location. Well, fluorescence is used to determine protein location in vivo using GFP, or green fluorescent protein, tagged to a cDNA library. So GFP stands for green fluorescent protein, and these green fluorescent proteins, you can use recombinant DNA technologies to stick a GFP sequence to the protein that you're interested in. And you can visualize it with a fluorescent microscope. So here we have a particular gene that we're interested in, the cDNA for that gene, and then we also have this sequence for your green fluorescent protein right after that. Okay, so in this case, when we're looking at under a fluorescent microscope in this cell, everywhere that's glowing green has that protein that has the green fluorescent protein attached to it. Okay. And GFP is really an awesome technique. This is the structure of green fluorescent protein. It makes what's known as a beta barrel, and then the certain amino acid residues there in the center of that barrel coming in close contact, and they actually make a fluorescent moiety in that beta barrel. And these can be tuned to give almost every color in the rainbow. Okay, so there's many different colors of these now. Okay, and so what that helps is you can actually label more than one protein at the same time and be able to see multiple protein locations. Or if they are in the same location, you would see kind of a mixture of those two colors. Right, so if we have the examples are shown here. Green is a glutamate receptor, purple is fat droplets here. In this picture we have red is the membrane, and green is a plasmid binding protein. If you have a case where you have overlapping signals, let's say you had green and red, two different proteins, and they were in the same location, then you'd get something that looks sort of yellow. Okay. Another way of visualizing where a protein is immunofluorescence. In this technique, you bind a protein with a primary antibody and then detect with a fluorescent secondary antibody. Okay. That's the most common way to do it. The other way is your protein is fused to a tag, and then the fluorescent primary antibody detects that tag. Okay. You can do this with several proteins in the same sample and just use different colors. Okay. If we look at this figure, this is our protein. Okay, we can have a primary antibody that binds to our protein, and then a secondary antibody that binds to the primary antibody, and the secondary antibody contains a fluorescent dye. Okay, so that's kind of the picture here, right? We have DNA polymerase here in red, another protein pCNA in green, bird U in blue, and then you can merge all of these images together and see where they're being co-localizing. So the proteins, when those signals overlap, those proteins are in the same location. Okay. This technique only shows that they're in the same location. It doesn't necessarily mean that they are interacting with one another. And sometimes protein-protein interactions are important and you want to study them. So ways to do that, ways to identify protein-protein interactions. You can isolate a protein complex. Okay. You can use a tag, an epitope tag, so something that an antibody would recognize from one protein in the complex, and then you gently isolate the epitope tagged protein, and that will also isolate any proteins that are stably interacting with that protein. These are often weak interactions, but they hold proteins together in complexes. Okay, all proteins isolated can then be separated and identified further. And this is what's referred to as TAP, or tandem affinity purification tags, enhance the procedure. Right, and this is yet another role of protein fusion tags. And then we also talk about the yeast to hybrid model with this technique. Okay, so for TAP tags, the first step is co-immunoprecipitation, or co-IP. So immunoprecipitation, you'll have your gene of interest, represented here as just cDNA from that gene, attached to an epitope tag. And remember, all epitope means is that an epitope is a sequence that an antibody binds to. Okay. So you express this tagged protein in a cell. Okay, then you get that cell extract, and you precipitate that tagged protein with a specific antibody. So you have an antibody attached to like some sort of a bead or something that can be separated from the solution. So your protein with the epitope tag will bind to that bead and be pulled out of solution. And that's what's known as immunoprecipitate. So if we look here, this is our pure protein that we're interested in, that's tagged. The immunoprecipitate that's pulled out of solution, we have that protein, but we also have 3 other proteins here. And those proteins are coming out of solution because they're interacting with that protein that has the tag on it. Okay, so in this case, co-immunoprecipitation, or co-IP, we're pulling out a protein and we're seeing the proteins that are interacting with it. Okay. The second procedure for doing this is what's known as TAP tagged proteins. Okay, a TAP tag, here are target proteins in red, and then our TAP tag is shown here in purple. And there's a few different parts to it. Calmodulin binding peptide, TEV, protease cleavage site. This site just allows us to cut off this piece, and then we have protein A at the end. Okay, so our crude cell extract contains proteins that interact with our target protein. Okay. We run this cell extract over an affinity column that binds to protein A. So IgG is an antibody, immunoglobulin, that binds to protein A, and this is a very strong interaction. Okay. Proteins that don't interact with this column get washed away, and then all we're left with is our protein of interest and the proteins that bind to that protein of interest. Okay, the next step is cutting that cleavage site, the protease cleavage site, and what we're left with is our protein and the proteins that bind it. Okay, then remember we also have this calmodulin site there. We can bind it to a column that has calmodulin beads and then immobilize our target protein, and then we can elute the loosely bound proteins. And sort of, if we do this sequentially, we can kind of see which proteins are loosely bound, and then we can elute our protein of interest and the strongly bound interaction, the proteins that are more strongly bound to it, by adding excess calmodulin to that column. Okay. The final method of identifying protein-protein interactions is the yeast to hybrid. Okay, in this method, your protein of interest is tagged with the Gal4 activation domain, when we can refer to that as protein Y. DNA library with all yeast genes is tagged with Gal4 binding domain, which we call protein X. And then you have a reporter gene under the control of Gal4. Okay. Differently tagged proteins must interact in order to get expression of the reporter gene. So whatever gene you have as your reporter that's in the control of Gal4, to get that, you have to have your protein Y and protein X interacting. Right, so this experiment's asking, does protein Y interact with protein X? So how does this work? Okay, so here we have our protein X, and it's bound to our Gal4 binding domain, which binds to our Gal4 binding site in the DNA. Okay. And our reporter gene is fairly close to this site. Next, we have our Gal4 activation domain bound to our protein Y. Okay. And when protein Y and protein X interact, what that does is that brings this activation domain very close in proximity, and now our RNA polymerase can form here, and then that reporter gene is going to be transcribed, expressed. Okay. The reporter gene is often one needed for either survival, or it's a gene that will color the yeast. Okay. Now, if we look at what happens here, if we have the yeast strain 1 with a Gal4 binding domain fusions and yeast strain 2 with Gal4P activation domains, you would mate them together to produce diploid cells that contain both, and then you plate on medium, requiring the interaction of those binding domains for survival. Okay, the survivors will form colonies, and then you can sequence the fusion proteins to identify which proteins are interacting. Okay, and so this is really not a technique that's used very much. It's in the book, so I just wanted to include it in the lecture. I probably would not ask a question about the specifics of this yeast 2 hybrid model, but I wanted to include it just to be complete. Okay. Right. So that ends up our semester. Just a reminder: the next class period will be our last in-class activity. We're going to go through designing some restriction sites on some plasmids. Okay, and then the final exam, another reminder, the final exam is cumulative. You can use a one-page sheet of notes on the final, so hopefully that helps. There's a lot of information we covered this semester, and I think it's kind of silly to have you try to memorize all of that material, so you can use a note sheet on that.